Very good morning, dear students. As we have discussed in the previous class, the next topic mentioned, that is the next unit mentioned in your syllabus, unit number 4, this is exclusively for the most important types of protein, that is known as enzymes. And these enzymes, as we have discussed, they are biological catalysts. Without enzymes, there is no biological process are happening. They have a lot of difference from the conventional chemical catalyst, and all the major salient features of the enzymes and introductory concept we have discussed in the previous class. And in this unit, the first topic mentioned in your syllabus, general properties of enzymes. So, enzymes are biological catalysts. They are proteins. They are soluble. They are globular proteins, and they have a perform characteristic function in our body. And we are going to discuss the basic aspects of the properties of the enzyme. So, the protein nature of enzyme, it was very important concept. Earlier scientific community studied a lot of aspects and they concluded that most of the enzymes are proteins. So, enzymes are a protein with catalytic properties due to its power of specific activation or specific activity. The enzymes are protein. They have a specific catalytic property, that is mainly because of their power of a specific activation to a particular reactions. And there are lot of evidence that provides the proteins nature of the enzymes. There are lot of concepts that clearly reveals that the enzymes are proteins. Some of the most important concepts are, the first one is the elementary composition. And in enzyme, the usual composition of proteins are carbon, hydrogen, nitrogen and sulfur. We have seen that how sulfur is present, the tertiary structure of in the proteins. Carbon and hydrogen and nitrogen, because protein consisting of amino acids, okay, that is nitrogen containing component, and as found in the protein. So the usual proposition or composition of the protein is carbon, hydrogen, nitrogen and sulfur. So these are the major elementary composition of the protein. And the next evidence in which that says protein nature of the enzymes are identical action of the enzyme over other enzymes and proteins. Similar action of some enzymes over other enzymes and proteins. The enzymes are subjected to the action of those enzymes which are specifically used for the breakdown of peptide bonds of proteins. Okay? The enzymes that are subjected to the action of the specific enzymes which are used for the breakdown of peptide bonds. Say for example proteases or peptidases. Okay? They are having identical actions over the other enzymes and the protein. The peptidases which cleaves the peptide bond, the breakdown of peptide bond. The third one is the amphoteric nature. This is very important aspects in which we can conclude enzymes are proteins. Okay, the enzymes behaves as the ampholyte in an electric field. Okay, enzymes that can behave as an amphoteric nature or ampholyte in an electric field. What are ampholyte? A components that has an acidic and basic groups, and they can form sweeter ions are known as ampholyte. We have already discussed in the previous unit Amino acids are the basic building blocks of proteins. The amino acid can exist, that has both the amino terminal end and the carboxy terminal end. One end is highly acidic and another end is highly basic. And in appropriate conditions, they can exist as a ionic form, that is sweeter ion, okay, under the influence of an electric field. That is why it is known as an amphoteric nature. Next one is denaturation. We have already discussed the major parameter that illustrate the change in the polypeptide chain. The folding of the polypeptide chain is the denaturation. All the protein they are undergo denaturation when the temperature is increased approximately 60 to 70. Then the hydrogen bonds or weak interaction will be broken and enzyme will be losing the function. And that is why that is known as it is known as denaturation. It is also caused due to the denaturing chemicals, and they destroy the activity of the enzymes. That means to say they destroy the three-dimensional structure, thereby destroy the activity of the enzymes. The percentage of denatured protein is mainly seen to be equal to the percentage loss in the enzyme activity. That is, the percentage of denatured protein, how much ever the percentage of denaturation is present, that much percentage loss of enzyme activity is studied. That is the another important aspect. Next one is the formation of antibiotics. Okay, so the theory suggests that the enzymes are proteins. Many purified enzymes on injection to the animal cells. Once we inject the purified proteins into the animal cells, they produce specific antibodies. Okay, many purified enzymes on the injection, they produce specific antibodies or immunoglobulins. We have seen the specific aspects of immunoglobulins. So these are the main aspects they say proteins are the enzymes. That is the elementary composition, the identical action of some enzymes other than that in comparison with other enzymes. Then the ampholytic or amphoteric nature, the denaturation capacity, and the formation of antibodies or immunoglobulins. Now, what are the chemical features of enzymes? They are simple protein enzymes. So that is classified into the chemical nature of enzymes are either they are simple protein or they are complex protein. Then why they are simple protein? Because they consist of simple protein. Only simple protein, example urease, amylases, papain. Okay, they consisting of varying composition of linear amino acid. They are very simple amino acid. They are able to perform a characteristic function. Urease, they are able to cleave urea. Amylase, they are able to cleave starch, and papain. Okay, they are able to cleave the proteins. Okay, so these are some of the important enzymes. Then the complex protein enzymes, these contains more conjugated protein. They are not simple. They have the combined proteins. They have both the protein and the non-protein part that is present in the enzymes. In the structure, we will see that. The protein part of the enzyme is known as apoenzymes, and the non-protein part of the enzyme is known as prosthetic group, which are generally associated with the protein. Okay, and both the apoenzymes and prosthetic groups are known as the complete enzymes, that is known as hollow enzymes. So that is why I told you the protein is a very complex molecule that consisting of a protein part apoenzymes, non-protein part the prosthetic group, that is commonly known as a hollow enzymes. So these are the chemical nature of the enzyme. Now what are the characteristic properties of the enzymes? Based on the theories in which they are talking about enzymes are protein, and based on the characteristic nature of the protein, the main important features of the protein, okay, or enzymes are, we are going to discuss one by one. They are colloidal in nature. Okay, the enzymes are molecules are of giant size. Okay, the enzymes are big molecules and their molecular weight is 12,000 to 1 million. Okay, they are completely colloids. Okay, their colloids are the mixture of substance that consisting of salts, gels, okay, and foams. Okay, these enzymes are mainly a colloidal substance that has got a salts and the gels. Then the next one is the catalytic nature or the effectiveness of enzymes. The catalytic property, that is the enzymatic activity. So the universal feature of all the enzymatic reaction is the absence of any side products. So enzymatic reaction, they will not give any side product. That is what I told you there. The catalytic activity is so special and so specific. They are so effective in doing the catalytic property. That is why we will not get any end product as a side product. Enzyme is adapted to the catalyze a very particular reaction. Okay, one particular enzyme is very specific to one particular enzymatic reaction. The catalytic property of the enzyme is very high in comparison with the other chemical catalyst. The catalytic power of an enzyme is estimated or identified or measured by turnover number or molecular activity. Okay, the catalytic potential or catalytic power of an enzyme is identified or measured in terms of the turnover number or molecular activity. So what do you mean by this molecular activity or turnover number? So turnover number or molecular activity may be defined as, this is the number of substrate molecule converted into a product per unit time when the enzyme is fully saturated with the substrate. When the enzyme is fully balanced with the probable substrate, then the number of substrate molecule convert it into a product per unit time is known as the turnover number or molecular activity. This is very important aspects in the case of enzymatic property. For most of the enzyme, the turnover number falls between 1 to 10 raised to 4 per second. This is the standard estimate. In most of the enzyme, the molecular activity or turnover number, that is between 1 to 10 raised to 4 or 1 to 10,000 per second. Then the next feature is the specificity of the enzyme action. This is very important. Enzymes are so specific. Enzymes are specific in their actions, so they act on a particular type of chemical reaction. The specificity is mainly depending upon the following factor. Number one, on one specific type of substrate. Because enzymes specifically bind to a substrate, which type of substrate it is going to bind, the enzyme is also changed. Then the group of structurally related compound. I told you the structure is very important. The structure is almost similar. The three-dimensional structure is almost similar. The enzyme in which that is going to act, this substrate are almost similar. Then the next one is only one of the two optical isomers of the component. Okay, the enzymatic activity will happen. One of the two optical isomers, okay, it will not active against both D and L variety. You know that there are two isomeric forms, the dextrorotatory and levorotatory. The enzyme will not act on both levo and dextrorotatory. It will act only in one particular forms, either levo or dextro varieties. And the next one is the enzymes on only one of the two geometrical isomers. The enzymes act on only two geometrical isomers. So you know that there are two types of geometrical isomers, cis isomers and trans isomers. So the enzymes will not act both the geometrical isomers such as cis and trans variety. They act only either cis or a trans. Most of the cases they are acting on the trans variety. Then in this particular case, we will discuss one by one the absolute specificity of the enzyme. That is for the first point, the enzyme is specific to one particular substrate. So here, some enzymes are capable of acting only one particular substrate and they will be catalyzing the chemical reaction. So for example, the enzyme urease, in microbiological point of view, you are having Very, very important aspect, because urease test is identified as one of the most important enzymatic reaction to the identification of the bacteria. So urease act on the urea, which cleaves urea and produce ammonia and carbon dioxide. Okay, that is the action of urease. So urea will not act on any, it will not bind to any other particular component other than urea. Okay, that's what I told you. The substrate of urease is urea, and it produces ammonia and carbon dioxide. If the urease is bind to urease, then only ammonia and carbon dioxide will produce. It will not go and bind to any other substrate. Second one, the group structurally related component, the group specificity. Okay, some other enzymes are capable of catalyzing the reaction of a structurally related components. Okay, the structures are the same, almost the three-dimensional structures are the same, then we are knowing that the functions are also same. Okay, such kind of enzymes are known as isoenzymes. So the isoenzymes are the enzyme that is structurally they are similar, but they are acting on the almost the same functionally related component. For example, lactate dehydrogenase or LDH, they catalyze the interconversion of pyruvic acid and lactic acid and a number of other structure related components. So lactic acid dehydrogenase or lactic dehydrogenase, they generally catalyze the conversion of pyruvic acid to the lactic acid, but they are not only do pyruvic acid and lactic acid like urease, they act on similar structurally related components, and this enzyme will be acting on this kind of components. So that is a group specificity. The next one is the enzymes act on only one of the two optical isomers. Okay, the optical specificity is very, very important. Particular enzymes will react with only one of the two optical isomers. I told you the optical isomers, the activity of an enzyme with a plain polarized light. Okay, so 50% of the molecule will be rotating as L variety and 50% of the molecules will be rotated as the D variety. Say for example, arginase, an enzyme that specifically act on arginine, you know that. But which arginine? The isomer should be always, the optical isomer should be always L variety. It will not act on the D variety. Okay, so arginase act only on a L variety. Similarly, D amino acid oxidase, which oxidize only D amino acid, that will not oxidize L amino acid. Okay, so that is what is that is the D amino acids are only keto acids. That is a specificity. Okay, the optical specificity of the amino acid. Then the next one is the enzymes are act on only one of the two geometrical isomers. Okay, so I told you the geometrical specificity, geometrical isomerism, either cis or trans. Okay, so some enzymes exhibit the specificity towards the cis and trans forms. Okay, so generally the enzymes, generally they act only cis forms or trans form, but they very rarely act on the cis and trans form. But there are some enzymes that act on the cis and trans form. Example, fumarase, they can catalyze the interconversion of fumaric acid and malic acid. So malic acid and fumaric acids are the isomeric forms of the components. Okay, they are isomeric form. One is a cis, another one is trans form. Okay, that is one of malic acid and fumaric acid. So fumarase can catalyze these two reactions. Okay, so that is what is the optical specificity. So based on the specific activity of the enzyme, the absolute specificity or group specificity, the optical specificity and geometrical specificities are very, very important. The next one is the thermostability or heat sensitivity. So you know that the temperature is very, very important parameter for the activity of the enzymes. So enzymes are very sensitive to heat. So as you all know, enzymes are protein. So they are having an optimum temperature for the ideal activity of an enzymes or protein. If you increase the temperature, what will happen? The specific activity, the enzymes will be getting denatured due to the heat accumulation. The tertiary structure of the protein will be degraded or distorted, and the V contraction will be destroyed. They they lack the function. The polypeptide will be completely unwind to the native structure, to the the unnative structure. Okay. So the rate of enzyme action increases with the rise in temperature. Generally, the temperature is increases, the activity of an enzyme is increases. But once it is reaches a specific temperature, if you still increase the temperature, the activity decreases. Okay, that's what I told you. The enzymes are thermostable. Okay, thermolabile. Okay, sorry, not thermostable, thermolabile. They're heat sensitive. Okay, they're not thermostable, they're thermolabile. Okay, please note this. Okay, I'm sorry, extremely sorry for the confusion. I think I have told thermostability, this thermolabile. So each and enzyme require optimum temperature for the ideal functioning of the enzymes. Okay, so once it is getting the ideal temperature, if you increase it still more, then the activity decreases. That is what I told you. Generally, if you increase a temperature, it will the enzyme activity also increases, but until it reaches the optimum temperature. Okay, so after the optimum temperature, if you further increase the temperature, the activity will decrease. So that is the importance of thermolability or heat sensitivity. They are very, very sensitive to heat. Next one is the reversibility of a reaction. Okay, the next feature is the reversibility of a reaction. The enzymes are capable of bringing reversion in a chemical reaction. They are able to do both the sides. Okay, in equilibrium. Okay, so the digestive enzymes catalyze the hydrolytic reaction which are reversible. Okay, so many digestive enzymes they are in our body, that is in intestine or gastric areas or stomach. Okay, all there are lot of digestive enzymes, they catalyze the hydrolytic reaction that are reversible. Okay, say for example lipases, they catalyze the synthesis of fats from glycerol and fatty acid, and once the hydrolysis happen, they convert the component their individual unit. Okay, we have discussed that is in the this particular fact. Okay, unit of lipids. So you know that triglycerides are the combination of glycerol and fatty acid. Okay, that's not triglycerides. So when this triglyceride, so that is catalyzed by lipase, the reversible reaction when triglycerides are interacted with the lipase, so first what will happen? It will be produce the diglycerides, then fatty acid. Then again if you interact with the next lipase, then the monoglycerides will be produced plus 2 fatty acid. Then finally if you react with one more lipase, what will happen? The lipase will go and act the primary position of the carbon atom, and you will be able to produce glycerol and 3 molecules of fatty acids. Okay, so that is what is the importance of the reversibility. Both the reaction they can catalyze, they can form triglycerides and they can break down triglycerides. Okay, then the next one is the sensitivity of hydrogen ion concentration. Okay, the pH. Okay, the pH value or the hydrogen ion concentration of the medium controls the activity of the enzymes. So the pH value, like temperature, if the pH is increased, the enzyme activity also increases. But once the optimum pH is reached, the enzymatic reaction will be getting stabilized, but still increase in the pH will decrease the enzymatic activity. So the related to the degree of dissociation to the electric charge of the enzyme to the formation of an enzyme and substrate. Okay, the pH sensitivity, they relate to the degree of dissociation to the electric charge of the enzyme to the formation of enzyme or the substrate complex. So the hydrogen ion concentration depend on mainly the following factors, such as the nature of the buffer system. Okay, you have already discussed in the unit number 1. Buffer is a chemical mixture that resists the change in pH when you are adding acid or alkali. That is known as buffer. Okay, so the nature of buffer system. Next one is the presence of other colloids, activators or inhibitors. Okay, other colloidal substance, there are certain agents they will activate or there is certain agent they will inhibit. Okay, so all these activators, inhibitors and colloids are the main agent that adversely affect the hydrogen ion concentration of the enzymatic activity. Then the age of the cell or tissue in which that is happening. Okay, the age of the cell or tissues. Okay, the mature cells or the newly growing cells. That is very, very important. Then the nature of the substrate. I have already told you the enzymatic activity is very specific to the substrate. Based on the nature of the substrate, the pH is also plays a very, very vital role. Then in general, the maximum enzymatic activity is obtained at the near the isoelectric point of the enzyme. So enzymes are protein. They are able to produce both the amino terminal groups as well as carboxyl terminal group under appropriate pH. The hydroxyl group that is from the carboxylic acid group, the hydrogen will go and attach to the NH3 end and it will become sweeter ion. NH3 plus and CH3 or COO minus. Okay, that is known as sweeter ion. Okay, so once this sweeter ion is produced, that is neutral in charge. The pH at which the amino acid get neutral in charge is known as a isoelectric point. This we have already discussed. And the maximum activity of the enzyme, this we can observe in the isoelectric point. So these are the main important characteristic features. So we have discussed the main characteristic features: the colloidal nature, the catalytic power of the enzymes or effectiveness, the specificity of the enzymes such as absolute specificity, group specificity, optical specificity, geometric specificity, then the thermolability or heat sensitivity, then the reversibility of the reaction. Next one is the hydrogen ion concentration, sensitivity or pH sensitivity. So these are the major characteristic features of the biological catalyst or enzymes. Dear students, now we can conclude today's class. We have discussed what is the general, what do you mean by protein, that we have already studied, and enzymes are protein. There are a lot of the theories they says that the reason we are saying that enzymes are protein. What are the evidence? The major evidence that says general properties of enzyme, the proteinaceous nature of the enzyme, their elementary composition, identical action of the enzymes over other enzymes and protein, ampholytic or amphoteric nature of proteins, denaturation and formation of antibodies. These all the aspects will clearly tells about enzymes are proteins. Then what are the chemical nature of the enzymes? Based on the chemical nature, they are simple proteins. They are having only simple basic amino acid units. Then or complex protein, they have both the protein part and non-protein part. The protein part is known as a apoenzyme. Non-protein part is known as coenzymes. And the prosthetic group and the complete part is known as a holoenzymes. Then what are the characteristic features of the enzymes? Their colloidal nature, the catalytic nature or effectiveness, the specificity of the enzymatic action, thermolability or heat sensitivity, reversibility of reaction, and the pH sensitivity. So these are the important concepts we have discussed in this particular class. So these are the main important textbook, dear students. Whatever I have taken, it is there in the Principles of Biochemistry by Leninger, or Fundamentals of Biochemistry by Jane, or any of this particular textbook, especially Biochemistry by Voigt and Voigt, you can refer. Okay. So these are the most important concept in this particular topics. So thank you very much for this watching this particular class. If you are having any clarification, you can write it to me or you can ask me. Thank you, dear students. Thank you very much.